So today we have Christina Annunziata talking. She's an investigator in the Women's Malignancy branch. She got her MD at Georgetown University Medical School, and she did her internal medicine training, and she came to NCI in the medical oncology branch, working with Lou Stout, investigating NF-kappa B signaling in multiple myeloma. And subsequently, she moved to the medical oncology branch, and she started investigating NF-kappa B in ovarian cancer. And she has another responsibility. She oversees the clinical trials in the women's malignancy branch. So the title of her talk is Ovarian Cancer in the Genomics Era. Tina.
Okay, great. Thank you very much. And this is on. So this is my, I guess I've done this lecture for several years, and it's kind of morphed over the last couple of years. So today I wanted to introduce genomics in medicine and also genomics in as it applies to ovarian cancer. So what is genomics? It's defined as the study of the genome, and usually it involves studying chromosomes, gene expression, and really studying the global analysis of these things and not just individual entities. So the genomics era, if you think of it that way, actually started back in 1959 with the original discovery by Noel and Hungerford, who were studying chromosomes in leukemia to identify a recurrent abnormality, which they called the Philadelphia chromosome, because that's where they were located. And they found that this occurred in a type of chronic leukemia. So they looked at just karyotypes and found that here, as you could see in this arrow, this particular chromosome was not normal. And that occurred in pretty much all patients with a form of chronic leukemia. And that was back in 1959. And so then it was not until 1973 that Janet Rowley looked at the karyotypes and found that this was actually, there was a recurrent loss of 22q in all these patients. Then it was not until 1984 that Goff et al. Found that this was actually joining between the chromosome 22 and a fusion between what he called breakpoint cluster region, BCR, and the gene ABL, the ABL kinase downstream of that. So then in 1996, so this is, from 1959 until 1996, we finally came up with a drug that blocks the constitutive ABL kinase activity. And that was by Brian Drucker in 1996, showed that this drug, CGP57148, actually specifically block the ABL kinase and can be used in these patients with the chromosomal abnormality to block their ABL-driven leukemia. And so that was a very long process, and now eventually this became approved as the drug imatinib, which is given for patients with CML. And that completely transformed the treatment of CML, of chronic myelogenous leukemia, where before it was incurable, and now patients are essentially, most patients can achieve long-term remissions. So that was an observational genomics. So what can we do to speed up that timeline a bit? And so people have looked at something which we call functional genomics. So now we're looking at not just what part of the genome is abnormal, but what part of the genome is functional, actually causing the cancer, causing an effect. And what actually transforms normal cells into cancer. So now we're looking for what is currently termed driver alterations. So in 1981, kind of the first functional genomics experiment in the cancer world occurred, and this led to the discovery of the oncogene HER2/neu, which some of you may know as in breast cancer, or certain types of breast cancer. So what they did was they actually looked in carcinomas and neuroblastomas to look for genes that would transform normal mouse fibroblasts. So they basically took carcinomas and neuroblastomas, and that's where the "neu" comes from neuroblastomas, to see, they chopped up the genome and basically transformed it into mouse fibroblasts and saw what made the mouse fibroblast able to then grow as a cancer in a mouse. So that was in 1981, and now in 1984, Schechter et al. Found that this actually was a recurrent abnormality that was that they termed new, and found that the new actually had similarities to EGFR, which is epidermal growth factor receptor. And this is a southern blot showing these ERB-B related sequences. Then in 1985, Cousins et al. Discovered that this HER2, so it's called HER2 and new, so she called it HER2, so human epidermal growth factor receptor 2. She called it HER2, and it found that it was recurrently amplified on chromosome 17. And then in 87, Dennis Slayman et al. Found that this chromosome 17 by southern blot was amplified in a subset of breast cancers. And so this discovery led to an antibody against this HER2 protein, which is called Herceptin, which is used in a subset of breast cancers that overexpress, that have this HER2 amplification. So that was a slightly quicker timeline. So we have used genomics in ovarian cancer to ask if we have any drivers in ovarian cancer. But first, let me talk about ovarian cancer, since this lecture is on ovarian cancer. Ovarian cancer is the most lethal gynecologic malignancy in the United States. It leads to greater than 16,000 deaths per year, and it's the 5th most common cause of cancer death for women. It's diagnosed, 70% of women are diagnosed with advanced disease, which is why it makes it such a deadly disease. Less than 35% of the patients with advanced stage disease are alive at 5 years. So what is advanced stage disease? Advanced stage, we typically term stage 3 and stage 4. So stage 1, you can see, is actually quite curable by surgery, and a surgery alone in most cases. That's when the cancer is found confined to the ovaries. When cancer is confined to the pelvis, you do have a pretty good surgical cure. But when it's spread to the intraperitoneally or to the nodes, there's less of a chance of survival. And then, of course, if there's distant metastasis at the time of diagnosis, the survival is quite poor. So the treatment for newly diagnosed ovarian cancer in all cases is complete surgical staging. So when ovarian cancer is diagnosed based on imaging and a biopsy, you have to undergo complete surgical staging with optimal reductive surgery, meaning leaving less than 1 centimeter of disease. That is the goal, is to leave less, actually to get rid of all the disease, followed by chemotherapy, and of course, clinical trials, which is what we do at the National Cancer Institute. So complete surgical staging and optimal reductive surgery is a surgical talk. I'm going to focus on the chemotherapy that we do for ovarian cancer. And standard chemotherapy always involves a platinum and a taxane. So the platinum agent is typically cisplatin or carboplatin, and the taxane is usually paclitaxel, but can also be docetaxel if there are problems with the paclitaxel, which can cause allergic reactions in some cases. If the patient is diagnosed with stage 3 ovarian cancer and has had an optimal surgical reduction, which means less than 1 centimeter of cancer residual at the time of completion of the surgery, then that patient should undergo intraperitoneal administration of the chemotherapy. And that means the platinum, either cisplatin or carboplatin, is delivered into the peritoneal cavity, into the abdomen instead of IV. And the taxane is given both IV and IP, and that's because the taxane is a hydrophobic carrier, and it does not get absorbed from the peritoneal cavity into the IV. So we need to give the IV in order to get the full systemic exposure. The platinum is absorbed greater than 95% into the circulation. So when you deliver it IP, it does get into the circulation. So this is a timeline showing our survival rate based on the chemotherapies that were available for ovarian cancer. So in the 1960s, there was only alkylating chemotherapy. Those are sort of like the mustard gases that were discovered very early on that worked well for leukemias. did not work at all for ovarian cancer. There was no one who lived with advanced stage, which with the stage 3 or stage 4, no one lived past 5 years. With the introduction of cisplatin in the 1970s, there was an improvement to about 5% of patients who could live beyond 5 years. And then combination therapies with alkylators and cisplatin actually did bump the response rate up to, or the survival rate up to 15% in the 1980s. The big breakthrough was the discovery of the taxanes. The first one was paclitaxel in the 1990s, and that bumped the survival. The combination of paclitaxel and a platinum, either cisplatin or carboplatin, gave the survival back, jumped up to 35% in the 1990s. And then, as I mentioned, in the 2000s, the introduction of the intraperitoneal administration of the same chemotherapy has an additional improvement in overall survival out to 40%. So what is ovarian cancer? So ovarian cancer actually is quite a heterogeneous disease. We call it ovarian cancer because most of the time it's diagnosed at the point of giving a large mass on the ovary. But I'm going to talk about that in a little bit. The histology of ovarian cancer, you can see here, is quite variable. The serous cancers are the most common, and those are 80%. There can be endometrioid cancers, which look much more glandular, like they might have come from the uterus, and that is about 10%. Clear cell cancers, you can see down here in E, they look like on histology that they have clearing of the cytoplasm. Mucinous cancers, which look like GI, actually gastrointestinal cancers, and then other 2% is quite infrequent. So they look quite different, and so they probably don't all arise from the actual ovary. So the recent thinking actually is that serous cancers actually arise in the fallopian tube and not the ovary itself. However, when they pass through the fallopian tube and sit on the ovary, there's a rich hormonal environment there that stimulates the growth and honing of the cells to the ovary. Endometrioid ovarian cancer maybe maybe it comes from the endometrial cells, that the glandular cells that pass through the fallopian tube and then find a home on the ovary. This was linked to endometriosis in both endometrial and clear cell cancers. Actually, there is an epidemiological link, at least, to endometriosis. And then the mucinous cancers, some have proposed that they might come from endocervical glands, which actually produce a lot of mucin. Some people have proposed that they are misdiagnosed ovarian cancers and they're actually appendiceal cancers that then go sit on the ovary. So the mucinous cancers are quite a mystery, and we haven't quite figured out what to do, where they come from, and therefore how best to treat them. So that is an area of active research. So in ovarian cancer, of course, we believe that increasing our understanding about biological and biochemical events underlying cancer progression will create avenues for new treatments. So coming back to genomics, how can we use genomics to do this, to learn more about it? So I'll start with clear cell and endometrioid cancer. The clear cell cancers are about 5 to 10% of all cases. And as I mentioned, the serous cancers were 70 to 80%, so much less frequent. The clear cell cancers do tend to have a worse response to the standard platinum and taxane chemotherapy. And as I mentioned, up to 40% of clear cell cancers are associated with a diagnosis of a prior diagnosis of endometriosis. The genomics of ovarian cancer were actually, of clear cell ovarian cancer were actually, there were initial study was published in 2010, where they actually sequenced RNA from 18 clear cell ovarian cancers and 1 cell line in a discovery phase. Then they sequenced DNA exons from 210 samples, 101 more clear cells, 33 endometrioid ovarian cancers, 76 serous cancers, and 1 more clear cell line. And they did an immunostaining of 455 samples in a tissue microarray. So what they found was recurrent abnormalities in this gene, ARID1A, and that was tightly linked to the clear cell cancers. And as you can see, the mutations are dispersed throughout the entire gene. So they can have nonsense mutations, insertions and deletions, missense mutations, all throughout the gene. And they occurred in both clear cell and endometrioid cancers in the data set. So what is ARID1A? ARID1A is actually part of the SWI/SNF chromatin remodeling complex. It's mutated in breast cancer and in lung cancer, and it's on chromosome 1p36, which actually has been found to be deleted in 6% of all cancers. So given that it's deleted, it's most likely a tumor suppressor gene. ARID1A was lost, so BAF250A is the protein name for ARID1A. So this is correlating protein with genomics. So here's the sort of darker orange color showing that when the gene is mutated, 73% of those cases have full loss of expression of the BAF2A protein. And that's in clear cell cancers here and endometrioid cancers here. And as mentioned previously, well, I didn't mention previously, but there were no mutations or loss of proteins found in high-grade serous cancers. So this is telling us that high-grade serous cancers do have a genomically different setting than both clear cell and endometrioid cancers. Clear cell and endometrioid cancers appear to be genomically similar. High-grade serous cancers are genomically different, at least in this particular locus. So to summarize that major finding, ARID1A was mutated or lost in over 40% of clear cell cancers, 30% of endometrioid cancers, and less than 1% of serous cancers. However, there's an unknown oncogenic mechanism at the current time. There's no indication of which resulting pathways are affected, although now recently it's been thought that it's in the VEGF HIF1 alpha pathway, but that's currently under study. There's unclear therapeutic utility. So there potentially is diagnostic utility in this case, but because it was not a functional experiment, and it was sort of the first example which I showed you, which was more of an observational experiment, there's not a treatment implication at the current time, although people are looking into that. Mucinous cancers, as I mentioned, is very rare, but it has some genomic studies have been done on mucinous cancers, which I'll briefly tell you about. Mucinous cancers here in the dotted line, this is just overall survival and years since diagnosis. So compared to non-mucinous cancers, patients with mucinous ovarian cancers actually tend to have a worse overall survival. And that's mainly because it responds poorly to the standard chemotherapy regimens administered for traditional ovarian cancer. So back in 2006, there was a very early gene expression microarray study done with serous versus mucinous cancers. And you could see here that the clustering, I think I have a box around it, mucinous cancers clustered based on gene expression quite differently than the serous cancers. So here we have low-grade serous cancers, we have high-grade serous cancers, and we have mucinous low-grade and high-grade cancers here. So mucinous cancers are genomically very different than serous cancers as well. Not much is known about what genes are in there, although people are working on what signatures that are. People don't study mucinous cancers as much. There's not been as much study because of its rarity. This is a very, very small study showing that 50% of the cancers had a mutation in KRAS, which is a potentially interesting target that should be followed up. Low-grade serous cancers. So I mentioned serous cancers are 80% of ovarian cancer, but serous cancers actually can be divided into low-grade and high-grade based on their pathology, so based on how they look in the microscope. Low-grade serous cancers are actually probably a different origin and a different development pathway than high-grade serous cancers. So KRAS and BRAF are also interesting in low-grade serous cancers. There was a study back in 2003 that looked at KRAS and BRAF mutations in low-grade serous cancers. and found that 68% of low-grade serous cancers actually had mutations. 61% of 68% of low-grade serous, and this is borderline tumors, which is actually considered a precursor lesion to low-grade serous cancers. None of high-grade serous cancers had a KRAS mutation. So that's also pointing to a different origin. And this is just showing you graphically that serous borderline tumors, which is a precursor lesion, and invasive low-grade serous cancers have a relatively higher expression of KRAS and BRAF, mutations of KRAS and BRAF, but high-grade serous cancers have 0. So the RAS signaling pathway is an interesting pathway that could potentially be targeted in low-grade serous cancers. This is a very sort of low-res, simplistic diagram of RAS signaling. So downstream of EGF receptor, you can have KRAS signals through RAF, MEK, and ERK to activate gene transcription, which promotes cell survival and resistance to chemotherapy. So the thought is that something downstream of RAS could be inhibited. And so MEK inhibitors were designed, and MEK inhibitors block downstream of RAS. Selumetinib is one particular MEK inhibitor that was tried in a clinical trial of low-grade patients with low-grade serous ovarian cancer. On this trial, 52 patients enrolled. 8 of them had responses, and 34 had stable disease greater than 4 months. But interestingly, when you look at the characteristics of the patients who responded, it was not really associated with the KRAS mutation. So in this graph, patients, this is a KRAS or BRAF mutation, patients who did not have, of the 18 patients who did not have a mutation, 5 of those responded, which was 28%. But only 2 of the 16 patients who did have a mutation responded. So what does that mean? So the KRAS mutation or the BRAF mutation was not predictive, really, of response to a MEK inhibitor. So is that because the MEK inhibitor didn't work, or is that because, as we know, RAS signals a lot of, so if RAS is mutated, this is really not the only pathway it's signaling through. So probably, most likely, there were compensatory pathways that overtook. So probably it was not that the RAS was a problem. I mean, the RAS probably was the problem, but the MEK inhibitor was not the best choice for signaling, for inhibiting signaling in KRAS mutant tumors. So more work is actively being done in low-grade serous cancers to figure out better targets. Okay, so now we get to high-grade serous cancers. So high-grade serous cancers are the most common of the ovarian cancers. They are genetically and genomically distinct from low-grade serous cancers. So in 2011, the original Cancer Genome Atlas study was published in Nature. This was a clinically annotated high-grade serous ovarian cancer samples, and the goal was to identify molecular abnormalities that could influence pathophysiology, affect outcome, and constitute therapeutic targets. What was published there at the time was 489 tumors that underwent microarray analysis using messenger RNA expression, microRNA expression, DNA copy number, and DNA promoter methylation. And at the time, they had a whole exome sequencing and the DNA for 316 samples. That has since been expanded, and if you go to the Cancer Genome Atlas portal, there are many more samples now added to this database. But let's go through what they showed in 2011. So the sample inclusion criteria here were only newly diagnosed patients, so no prior chemotherapy. Obviously, they had to have serous adenocarcinoma, no prior treatment. They had to have a companion normal tissue specimen. However, the normal tissue specimen did not need to be a normal ovary or normal ovarian epithelium. That could have been previously extracted germline DNA. So they needed to have something like that. So when you look at the TCGA database, keep in mind that your normals are only about, I think, 11 cases. Okay, so the first thing that that showed up is that high-grade serous ovarian cancer is genomically unstable. So this is actually quite interesting. So the first Cancer Genome Atlas data set that was published was glioblastoma, which is a cancer of the brain. And you could see here in this is a array CGH. So this is DNA chromosomes. So here you see chromosomes 1 through 22, and then this is the patient. Individual patients are sort of in the x-axis. And blue is deletion and red is amplification. So you can see in glioblastoma, white, most of it is white, so there's not really much going on compared to the normal DNA, except for this very bright here amplification and this very distinct deletion. In ovarian cancer, we have recurrent amplifications and deletions, but they're all over the genome. So there's a lot of craziness going on. It's completely genomically unstable. So that was kind of the main finding from here is that high-grade serous ovarian cancer is actually genomically unstable. Looking at mutated genes, there were some mutated genes, and the most common mutated gene was P53. So this also is a highlight of this study that high-grade serous cancers are actually probably require mutation or deletion of P53 in order to form. The other common mutations were BRCA1 and BRCA2, which I will focus on a little bit later in the talk. Even in patients with hereditary BRCA and BRCA1 and BRCA2, there needs to be a P53 deletion. So that always co-occurred. And then there were a number of other mutations, but far less common than hoped. So there was not a particular obvious driver in ovarian cancer other than P53. So if we focus a little bit more on the BRCA1 and the BRCA2, you could see that they were altered in about 33% of cases. And here the purple means mutated and the green is methylated. Sorry, the purple was germline mutation, the green was somatic mutation, and the gray is methylated, meaning silenced. But if you look at the difference between the mutated and the epigenetically silenced, actually the one that counts more is the mutation. So the patients with BRCA mutation actually do better. This is overall survival again. Patients with BRCA mutation actually do better in responding to chemotherapy and having overall survival compared to either the methylated or the BRCA wild type. So what is BRCA1 and BRCA2? They're actually in the homologous, this is HR, means homologous recombination pathway of DNA repair. And they found that 51% of cases were altered. However, really only the mutated ones, the 33% with the mutated, were important for survival. So BRCA1 and BRCA2, as I mentioned, are involved in the homologous recombination pathway of DNA repair, which also involves RAD51 and FANCD2, as well as potentially P10. So there were a number of pathways that were altered. The most common pathway was this FOXM1 pathway, and with respect to cell cycle progression and DNA repair, and that was thought to be a link. However, this hasn't really played out with respect to clinical application so far. So BRCA1 and BRCA2, CHEK2 is an interesting one that I'm going to talk about a little bit, RAD51 as well, and then cell cycle progression pathway is also under investigation. So what next? So how are we doing therapeutic approaches? How are we designing therapeutic approaches based on these findings? As I'm going to show you, 50% of patients may have a homologous recombination defect for the BRCA1 and 2 mutations, but other smaller, less common mutations as well. And I'm going to show you some work that we've done with PARP inhibitors. There's also some other commonly deregulated pathways such as RB and RAS and PI3 kinase pathway that might provide opportunities for treatment. But what we learned here is that there's genes or networks that are targetable and not necessarily individual driver genes, since the abnormalities occur throughout the genome. Okay, so I'm going to give you a little vignettes now about potential therapeutic strategies that we've discovered in high-grade serous cancer based on the TCGA and beyond. So BRCA1. So obviously BRCA is not new. If you've had a breast cancer talk, I don't know if you've had the breast cancer talk yet, but BRCA was discovered actually and cloned, not cloned, but discovered in 1990 by Mary Claire King. And it was discovered based on this pattern of breast and ovarian cancers that were found in families. So it was done by a traditional pedigree, lineage, and then linkage. But what we found from Cancer Genome Atlas is that not just BRCA1, but there are defects in the whole homologous recombination pathway in about half of the ovarian cancers. So there's BRCA germline mutation, there's BRCA somatic mutation. Methylation, as I showed you, does not seem to be as relevant. However, there's other homologous recombination defect proteins like RAD51, actually 51D and 51C can be mutated, P10 can be lost, and EMSY can be amplified. And all of these can lead to defects in homologous recombination, which probably is explaining why there is so much genomic instability, right? So if you can't repair these double-strand DNA breaks, you can get a lot of breaks that then get repaired incorrectly, and you can get a lot of fusions, amplifications, and deletions, explaining that genomic heterogeneity that we saw in the first TCGA slide. Beyond BRCA mutations, people are trying to look for not just mutations, but about gene expression patterns. And there's been a number of studies. This is just one example showing that we can find gene expression signatures of BRCA, of homologous recombination deficiency. And these expression signatures actually do show difference in survival, whereas the homologous recombination defective in blue has a better overall survival than homologous recombination intact. And in ovarian cancer cell lines, those with homologous recombination intact are less responsive to PARP inhibitors than ovarian cancer cells with deficient homologous recombination. Okay, so how does the PARP inhibitor work in these homologous recombination deficient cancers? So this is a DNA molecule and showing the example of a single strand break. So single strand breaks can occur naturally through exposure to cellular metabolism, environmental exposures, etc. The PARP enzyme is actually brought in to bind to the and mark the DNA that has a single strand break. So PARP stands for polyadenylation ribose phosphate. Polymerase. So a PARP inhibitor, if you have a PARP inhibitor, you can't repair the single strand break. And in replicating cells, when the replication complex gets to a single strand break, it actually causes a double strand break. So you have to unwind the DNA in order to replicate it. So when it unwinds, it causes a nick. But when you cause a nick in the DNA, at the point of a single strand break, it turns into a double strand break. The double strand breaks need to be repaired by homologous recombination. So the most efficient and the most intact way of repairing DNA, double-strand DNA breaks, is the homologous recombination pathway. However, in a BRCA deficient cell, if you've inhibited PARP, you overwhelm the cell with double-strand breaks, and there's no effective DNA repair. There are other pathways to repair double-strand breaks, but they are far less efficient and cause a lot of mutations in the DNA sequence. So in cancer cells with a BRCA deficiency, the hypothesis was the PARP inhibitor would overwhelm the cells with DNA breaks, double-strand breaks, and cause death. So olaparib is an orally active PARP inhibitor that we've done clinical trials with here at the NCI and have been, obviously done throughout the entire world. This is the schematic of olaparib, and this is just showing you that an example of where olaparib is actually effective in killing cells with BRCA1 mutation. This is a cell line, BRCA2 mutation but the cells with wild type BRCA1 and BRCA2 are resistant to killing with olaparib. So we did a phase 1 study with the combination of olaparib and carboplatin, and the thought was here that the carboplatin would sort of accelerate the DNA breaks, and if we could use that in combination with olaparib, then we would be able to sort of accelerate the whole process of killing the cancer cells. We had 3 cohorts here, and these have since been published. These are sort of old references, but these are now all published. This first publication was actually in the BRCA mutant cohort. Patients could have either breast or ovarian cancer with a BRCA mutation. We did another cohort with triple negative breast cancer, so that had normal BRCA, and we did another cohort with high-grade serous ovarian cancer that has also had a normal BRCA, with the thought that we could potentially augment the killing of the cells using the chemotherapy with the olaparib. We were able to dose escalate to the dose of olaparib 400 milligrams, which is the phase 2 dose, which is the regular dose of olaparib capsules twice daily for days 1 through 7, and the carboplatin was given at the full dose of AUC 5. In this cohort, we actually only a dose escalated to AUC of 4, so they were a little bit more sensitive to chemo, and in this cohort as well, probably because they had more prior lines of chemotherapy. So in the BRCA mutation cohort, this is sort of the genetically defined cancers, we enrolled 45 patients, 37 with ovarian and 8 with breast cancer. We had 30 patients in the dose escalation cohort and 15 in the expansion cohort where everybody got the same dose. The maximum tolerated dose was 8 carboplatin AUC of 5 on day 1 and olaparib on days 400 milligrams twice daily, days 1 through 7, and that was repeated every 21 days. So remarkably, this is a waterfall plot. So this is showing you response rates. So in a clinical trial, 20% growth in the up direction is considered progression. 30% decrease in tumor growth is considered a response, and anything obviously below that is a response. So anything below 30%. So this is color-coded by the dose levels, and dose level 6 is that maximum tolerated dose that I mentioned we did an expansion cohort in. But you can see here that in these patients with the BRCA mutations, we had a remarkable response rate. So more than half of the patients actually responded, and very, very few had tumor growth. And here's marked whether they had a BRCA1 or BRCA2 mutation. So even patients in the lower dose levels, of the carboplatin and the olaparib, actually had an ability to respond. So this is actually quite a response rate for patients who had so many prior chemotherapy regimens. There were no complete responses, even though it does look sort of like we had one complete response there. There was 15 patients, which is 44% of patients had a partial response, meaning 30% shrinkage or less. And then 38% of patients had stable disease greater than 4 months. And this is also remarkable, is that the patients were able to stay on study. So the response duration median was more than a year. This is also amazing, because usually, when you have a response, it comes and goes, within 6 months. But this is remarkable. So the median duration was 16 months and almost a year for the patients who had stable disease. We even did have patients out to 2 and 3 years. So the conclusion was that oral olaparib was well tolerated in combination with carboplatin chemotherapy, highly, highly active in advanced chemotherapy refractory BRCA deficient cancers, obviously greater activity at the higher doses, but this is positive proof of concept that the activity and tolerability of the genetically defined targeted therapy with olaparib in BRCA deficient cancers. So this is actually quite exciting because we have now a therapy that's designed for patients with a genetic abnormality. Okay, so that's one example of a success story for genomics. What about finding new targets? So where can we go with functional genomics in ovarian cancer? So here we're looking for things that can potentially be actionable. But just to step back for a minute, a lot of things have been called actionable that have not been linked to a functional experiment. So I don't know if you've heard about these tests like Caris or Foundation One, and they report that you can have possible or unlikely or likely benefit based on having a mutation. But these are not functional experiments. This is just sort of observational, again, sequencing. People have designed basket clinical trials based on this type of approach. So for example, we have the NCI impact trial where they're assigning treatments based on mutation. But as I mentioned, typically this is just finding the mutation and not pinning down the mutation as the actual functional driver. This is a very interesting article that was published a couple of years ago by Dr. Yanni and Reddig in Journal of Clinical Oncology. So they said the success of actionable mutations depends in large part on the strength of the data linking the target and the targeted therapy. For this trial design to work, two key combinations must be met. The tumor must depend on the target, and the targeted therapy must reliably inhibit the target. So I think this is where we are lacking. Achieving both goals can be a matter of some complexity. So this is actually very insightful because I think where we are lacking is that we haven't proven that things depend on the targeted pathway. So we hypothesized we need a functional experiment in order to do this. So we tried to do functional genomics in ovarian cancer. So we did a screen, a genomic screen, a functional genomic screen to identify targets. So here in this first experiment, we used an shRNA library where the shRNA are pooled, but they're barcoded, so we can pull them out later. This targets 2,500 human genes. There's actually now 12 to 15 shRNAs per construct, and they're all sequence verified. The main targets are, as expressed here, protein kinases, et cetera. So this library was originally published in 2006, but has been used many times since. So we did, so here's basically the layout for the experiment. You infect the cancer cells with the shRNA. This is kind of, well known now. Infect the cancer cells with the barcoded shRNAs. You can do an experiment where you induce the shRNA expression or you leave it off. And then in the induced shRNA, you look for things that dropped out compared to control. And these are things that are functionally important in the cells of interest. Amplify the barcodes, and actually we used to do a barcode microarray like 12 years ago, but now it's done by sequencing. So we did a functional genomic experiment in 4 ovarian cancer cell lines. We used 2 serous cell lines and 2 non-serous cell lines in order to look for targets. And actually we found, we looked for drivers in ovarian cancer in the serous versus the non-serous, or the pooled, or all of them. And if you look for things that were important in 3 out of 4 out of the cell lines to look for a reasonable amount of things to follow up on, we found 63 shRNAs representing 55 genes. And here's just a list of the 55 genes, but I will focus on one of them. We did follow up in 6 additional cell lines with 2 different RNAi constructs in order to validate this, and then we selected potentially druggable targets, meaning things that had drugs already in clinical trials. Focused functional screens, we are following up on combination matrices as well. So I wanted to focus on one of the genes that we have already taken, that we have taken to a clinical trial. So we found that CHEK1 was one of the genes that was actually overexpressed in nearly all ovarian cancers compared to normal controls. Our prioritization process, as I mentioned, was we started out with the 321 genes, narrowed that down to 65 genes, and then we looked in the Cancer Genome Atlas for things that were overexpressed in tumor compared to normal, and we found that actually CHEK1 was the most commonly overexpressed compared to normal. So what does CHEK do? CHEK is actually in the DNA damage and repair pathway, but it's a cell cycle modulator, not it modulates the cell cycle. So when DNA damage occurs, you have the DNA damage response, and in order to stop the cell cycle, in order to repair the DNA, this normally goes through P53 to stop the cell cycle in the G1S. Then before it gets to S, the DNA can be repaired, and then the cell can move on into synthesis and going to G2 and then M. But as I mentioned previously, in high-grade serous ovarian cancer, universally P53 is not functional. It's either mutated or lost. So cells can also go down the ATM pathway, which involves CHEK1 and CHEK2, in order to block the cell cycle at G2M. However, so this is probably why CHEK1 is amplified in ovarian cancers which have mutated P53, because they're trying again to stop the cell cycle in order to repair DNA before it can go into mitosis. So we thought if we use a CHEK inhibitor, it would be potentially synthetically lethal, or at least additively lethal with the P53 mutation. So we looked at the CHEK inhibitor in ovarian cancer cell lines. We published this back in 2014, where cells with high-grade serous with the P53 mutation were actually, they were more sensitive. This is just a cell survival curves. They were more sensitive, meaning they had a lower IC50 with the CHEK inhibitor compared to cells of other histology, which were more resistant to the CHEK inhibitor in vitro. We did obviously multiple additional experiments and eventually opened a clinical trial that is currently enrolling and is showing actually quite promising results in high-grade serous cancers without a BRCA mutation. And this was highlighted by a functional genomics approach. So here I'm going to show you, this is just initial data from our ongoing clinical trial. Here's patients with high-grade serous ovarian cancer without a BRCA mutation. Here's patients with the germline BRCA mutation. And as I mentioned, if this is the change in size of the tumor. If it goes above 20%, that's considered progression. If it goes below 30%, that's considered a response. So you can see all of our responses so far are in patients with the high-grade serous ovarian cancer without the BRCA mutation, as opposed to there's only stable disease or progression with patients with BRCA mutation. And then this is to point out that patients could have platinum-sensitive disease or platinum-resistant disease, and even the patients with platinum-resistant disease are also responding to this. So quite exciting. This is showing you the time on study. This is the patients who responded with the red circles and the time of their response, which was very early on in the first restaging, the second month. And you could see that many patients with the high-grade serous ovarian cancer are actually responding and staying on study for quite some time. The arrows mean that they're still on study. So this is quite an exciting trial as well. So because it came from our functional genomic screen where we found the CHEK1 and then used that in patients without a BRCA mutation where we thought that it would be most active, and it's quite successful. Okay, so to summarize, ovarian cancer is a heterogeneous disease. There are many different subtypes of ovarian cancer which have different genomic abnormalities. High-grade serous having the P53, the BRCA1 and 2, and we should add here the CHEK1 amplification or CHEK1 overexpression. Low-grade serous, even though it's called serous, is a different genomic disease. Mucinous, we still don't know what to do with. Clear cell and endometrioid have the ARID1A and the PI3 kinase, which I didn't talk about, in common. So we went from functional genomics in 1981 with the discovery of HER2. We've taken that now to a functional genomics screen with an shRNA library to identify drivers in the aberrant pathway, leading to a clinical trial and hopefully better treatment for women with ovarian cancer. And with that, this is the women's cancer team. The trial, two trials that I talked about are led by my colleague Jungmin Lee in the women's malignancies branch. Translational scientist who did the work on the Czech study in the lab. In my lab is Marianne Kim. These are the other members of my lab, and of course our collaborators. So with that, I will take any questions. Anybody?
With the PARP inhibitor, have you done any animal studies with that?
Yeah, so we've done, so the new things with the PARP inhibitor, actually, I didn't talk about, but we did in combination with the drug cediranib, which is actually a VEGF inhibitor, and that's also showing extremely promising results. That's actually in a national phase 3 study. But yeah, so we have done animal studies with various other combinations. Yes. Oh, so that's a really good question. So some of them, yes. So some of them tend to be more localized. So the mucinous cancers actually tend to grow to be really, really big in the ovary before they will metastasize. So I didn't talk about this, but one of the problems with ovarian cancer is that it's really hard to diagnose because there's not a lot of symptoms. And when there are symptoms, they're mistaken for like, diarrhea, constipation. They're mistaken for like gastrointestinal illnesses. So in order to have a symptom, you need to have a big mass. So the mucinous cancers do tend to be found more localized to the ovary compared to the other ones. Because they tend to shed less. It's very interesting how ovarian cancer will shed. The serous cancers shed very early. So they will be very small on the ovary, but they can be disseminated widely before the time of diagnosis. So that's another interesting pattern another interesting topic that I didn't talk about, which is the way ovarian cancer metastasizes, or it actually sheds into the abdominal cavity rather than sort of invading and going into the blood, like a lot of cancers. So cancers that metastasize via the blood tend to give you lung mets because that's the first place they'll go and lodge, lung, bone, brain, whereas ovarian cancer is localized to the abdominal cavity because it doesn't really invade early. It sheds before it invades. All right.
Our next speaker is Stephanie Goff, and she's in the surgery branch. And she got her first degree at Georgia Institute of Technology. And she got her MD at the University of South Florida. And she's been training under Steve Rosenberg here at NIH. She did her general surgery residency at Columbia University, followed by an oncology fellowship at Dana-Farber in Boston. And she's an associate research physician. Her title, Immune Checkpoint Blockade. Stephanie.
Hi, everybody. I'll make this a little bit informal since we're sort of a small group. But how many of you guys watch live television as opposed to like Hulu or Netflix or whatever? You actually turn on the TV and watch CBS. Anybody? So you've probably seen ads for Opdivo or Keytruda. Those are the drugs we're going to be talking about today. And so the story behind them is a little different than the pretty pictures of people sniffing flowers in the garden. But I'll tell you a little bit about how we got there and how these drugs have really made a change on the impact in the treatment of patients with cancer. So these are just our objectives for today, and hopefully we will meet all of them. One is just to describe the basics of immunotherapy, to describe sort of on a biological level what we think the mechanism of action of checkpoint blockade is. Some of the early clinical experience, which was actually here at the clinical center across the way, and the discovery of a new phenomenon for the FDA called immune-related adverse events. We'll go through sort of step-by-step checkpoint blockade in melanoma, only because it serves as a good model for checkpoint blockade in a lot of other cancers. And we'll talk specifically about the names will get more familiar to you as you see them: ipilimumab, nivolumab, and pembrolizumab. I will briefly touch on what data there are about checkpoint blockade in other cancers, and then look to some of the more experimental questions that are being asked about checkpoint blockade. So one important thing, as you might imagine, is that the treatment of patients with cancer is multi-pronged. It can't be held up by any one pillar alone. And so it's a combination of things that helps us treat these patients to the best of our ability. And surgery was the first, believe it or not. It actually dates back to Egyptian times. There's a scroll found from the 3000s BC where they show a listing of procedures and even hieroglyphics of a surgeon taking a tumor out of someone. I don't know they would have used the word surgeon, but it was clearly a little stick figure of a little man taking a little something out of somebody else. The next was actually radiation, and this came out around the turn of the century. Not shortly after Marie Curie figured out what X-rays were, they figured out that they could use them to burn tumors. And then recently, based on a lot of work done initially in Boston, and then the development of multi-drug regimens that happened across the street in the 70s at Building 10, was chemotherapy. And I didn't pick a 4-pillared STOA for no reason. It's that in the last few years, immunotherapy has become the 4th pillar in the treatment of patients with cancer. And it differs from the others in the way it works in the body. And we'll talk a little bit about that. So cancer immunotherapy, you can think of it in a couple of different ways. To use more generic terms, we can either turn on all the immune cells you have. So that's a non-specific stimulation of any potential immune reaction that's in your body. So one way to do that is to stimulate the cells that are there. We can do that with cytokines like IL-2 and IL-15. Another way to do that, instead of giving a positive signal to something, is to block anything that's getting a negative signal. So there's a lot of double negatives, and it's easy to get tripped up over the grammar and the language. But when you hear someone talk about checkpoint blockade, whether they say checkpoint inhibition, checkpoint inhibitor blockade, if you start to count up all the double negatives, it's easy to get lost in the meaning. But what everyone is referring to is that there is a negative influence on a cell, and you're stopping that negative influence. So you're taking the brakes off of that cell. So that's stimulating all the cells you've got. Another way is to teach the cells that you've got how to go after your cancer. And that's the second means, which is active immunization, which is a wide field of study in cancer vaccines. And the third way is not turning on all the cells you've got, not teaching all the cells you've got, but taking the cells out of your body, training them, growing them, creating them the way that you want, and then giving them back. And that's adoptive immunotherapy. Now, that's what my clinical specialty is. And we can speak more to that later if you'd like, but the focus of this is going to be checkpoint blockade. So what are some of the cells of the immune system? Once I got into my surgical training, it was very easy to forget that I had once known about B cells and T cells. But it's very fundamental and very basic what we're working on here, which is that B cells, which can make antibodies, T cells, which make helper CD4 cells, and cytotoxic or killer CD8 cells. This is where we think that checkpoint blockade is working, in that these T cells are being turned off for some reason by something in the microenvironment, by something in the tumor, by something in the patient, and if we can give the drug at the right time, it will turn them back on. So how is a T cell born? It's complicated, as you might imagine, and this is just the broad brush strokes. So you get a hematopoietic precursor that comes from the marrow, and it goes into the thymus, whether in a fetus or in a young adult, and it becomes designated to become a lymphocyte, a lymphoid progenitor. It goes through a stage which we call DN1, which is double negative. It doesn't show any signs of being a T cell just yet. It doesn't have a 4, it doesn't have an 8, but we know that one day it will end up there. It then goes into DN2, which just means it's gotten a little bit further down that path and can't be backed up. And again, 3 and 4 mean the same things. When you get here to double positive, what happens is that it connects with an epithelial cell and sees something. and gets trained to recognize something, and it becomes double positive. So it becomes specific for whatever it is here that it sees, but it hasn't determined yet which way it's going to go. But this is where it gets to be a little like Goldilocks, the story of Goldilocks. So if it doesn't get too much recognition at all, it dies from not being seen. Otherwise, it goes down and becomes either a CD8 or a CD4. And then here's where the second part of that Goldilocks fits in. If it gets recognized too much, if it's too strong, the body decides, hey, this might hurt us, we're going to get rid of it. So positive selection, even though the word seems good, positive and negative selection means that if something that gets hit with a stimulus that's too strong, it's going to get destroyed because it's going to be seen as dangerous. So what you end up with is a bunch of T cells in your body that recognize things to mild levels of degree, but not enough to hurt anything. And by doing this way, you can build a repertoire of T cells. And just to get an idea, there's about 4 × 10⁻11 T cells circulating in an adult human. If you take a thimbleful, there's about 1 million T cells in a thimbleful of blood. So there's a lot running around. A lot of them have their own unique specificity in T cell receptor. How do these T cells get activated? So we're going to walk through this a little slowly. Again, the figure is in your handout, and the reference for that handout is there, so you can read a little bit more. But what's important to know is that each T cell has a specificity. It has one or two things that sort of look similar that it knows how to latch into. And that's the first thing that has to happen for it to get activated. So this specificity is made up of 2 things. One, it's what the T cell knows how to see. And the other is what, quote unquote, white blood cell type the patient has. And that's because you've got these molecules called MHC, and that stands for major histocompatibility complex. You get half of these from your mom, half of these from your dad, and it takes these little pieces of antigen that have been chewed up by various proteasomes and mechanisms within the cell, and it puts it on the surface. So this combination of sort of pocket and ball has to be right for this T cell to see it. So the shape of the pocket is determined by just the HLA that you're born with, and you get that just like your A, B, or O blood type. And it really determines whether the CD8 can see it. So say I have a T cell in my body that recognizes a particular amino acid sequence, but it does it because I have HLA-A3. If for some reason, if we were doing adoptive cell studies and you took my T cell and gave it to somebody and they had that same sequence, but they weren't A3, that T cell would never know how to see it. It would just float around not doing anything because it couldn't see it. So it's the combination of the MHC and the antigen that makes this specificity. Now the specificity comes both in MHC1, which are on all nucleated cells, and it's a function of having a proteasome that's constantly chewing things up and recycling. It also comes from CD4 cells. And the reason this becomes important sort of from an evolutionary standpoint is that it takes in things to get endocytosed. So if you've got a bacteria, a virus, it'll get taken up by the cell, attached to MHC class 2, seen by a helper cell. That helper cell can then go off and tell a B cell to make an antibody. So this is how your vaccines against childhood diseases tend to work. You get fed these little bits of vaccine, and they go in and create this antibody. That's why you have titers against varicella or I'm blanking on one of the other titers, but the labs you get drawn to show that you've been successfully vaccinated. So it requires both the right blood type and the right antigen. But what else has to happen is that once that T cell sees its target, any of a number of things can happen on the inside of that T cell. And this is where the different checkpoints come into play. So this is the surface of the T cell. So all that we just talked about in the last slide was happening in the surface of a antigen presenting cell, and we weren't really talking about what happens in here. The next slide, we're talking about what happens in here once it engages here. So what can happen, anything that's black is a positive signal. So if it engages its TCR, so your little pocket has been filled, it's ready, it's gotten signal 1. If it engages CD28, that's positive. It goes down all these pathways, induces things in a positive way, makes that cell get activated. BTLA, also positive, turns on all these black pathways. However, PD1 and CTLA4 are negative pathways. They lead to blockade of RAS, blockade of AKT, blockade of PLC gamma. All these things shut the T cell down. And so in order to be activated, 1, it has to see its specific target, and 2, it has to be co-stimulated in the right fashion. The third part of getting a T cell activated is, well, okay, you've gotten your T cell, and it's gotten its co-stimulatory molecule here at CD28. What kind of T cell it turns into is a function of what other cytokines are around. So it's sort of, a cell will get polarized based on the cytokine profile present in the microenvironment. This is one area of interest at the moment because there's a way, if you can block both the checkpoint and get it past signal 2, can you augment signal 3 to make them more of a killer cell? But that's more of a research question right now. So what are the role of signal 2 checkpoints? So in a normal functioning human adult, human anything without cancer, the point of an immune checkpoint is to promote self-tolerance. So you may have T cells in your body that can see the lining of your heart, that can see the lining of your colon, that can see the tissues in your eye, the tissues in your skin. But because they're that Goldilocks middle, they're not doing anything bad. They're not going to harm you. And these checkpoints help keep them intact. What they do is limit collateral damage. And when that T cell recognizes something in the peripheral tissue, it will actually turn on all these checkpoints to stop it, because you don't want infection or inflammation just sort of running rampant. Your body needs to have a response to any sort of foreign antigen, but it also needs to be able to shut it down. And for cancer immunotherapy, what these checkpoints do, create the opportunity to break the tolerance to self-antigen. So what we're trying to do is stop the mechanisms that are keeping that in place. Because what if you've got a T cell that does know how to recognize a cancer, but it's being turned off? And there's a lot of double negatives here. But there are some really good reviews if you're interested in reading further at a time where you can sit back and think and go over things at your own pace. CTLA4 was the first one that we started to look at. So both naive and memory cells will express CD28 on the surface. That's a known positive co-stimulatory marker. However, if this gets bound really strongly, the cell knows it needs to have a way to shut off, so it starts to put CTLA4 on the surface. When it puts CTLA4 on the surface, you get a negative feedback that starts to shut it down. So it's its own sort of self-correcting mechanism. The stronger that binding, the more CTLA4, the more to turn it off. So it competes. It has the ability to compete because it has the same ligand as CD28. There are a special class of cells called T regulatory cells that this is their whole job, and they constitutively have CTLA4 on the surface. So they're always going around looking to shut down responses. PD1 works in a slightly different way. So once a T cell goes out to the peripheral tissue, engages with signal 1, and starts to be attacked, it upregulates the ligand for PD1. So if PD1 is present on the surface, it's geared up to stop this T cell, but it may or may not see the ligand. But as this tissue gets destroyed, it will upregulate that ligand, so it has a better chance of being seen. It's a really complex, really actually sort of beautiful when you think about it, way of self-regulating the system to come down. So it limits collateral damage during cell-mediated destruction of infection. So how can we take advantage of this for cancer? So there are some tumors that just by nature of the way that they have mutated, start to express PD-L1 all the time. And when that happens, these T cells don't have a chance. You know, as soon as they get to the cancer, they're going to see their ligand, they're going to get shut down. This happens infrequently, but when it does, it makes blocking this a target that seems reasonable. The other one is what I was talking about a little bit earlier. Once this gets engaged, and once this T cell starts to kill this cell, this realizes it. This T cell is making interferon gamma. When a tumor cell sees interferon gamma, it throws more PD-L1 on the surface. So eventually it gets enough momentum going and shuts down this T cell, which then stops this whole blockade, and things go back to normal. Again, if we could block this, if we could just let this interferon gamma hit this cell without any consequence, if this T cell could then destroy it, we could have a way to get around it. So where did we decide to start when we were looking into checkpoint blockade? Well, a good first place is to look at tumors that are known to respond to other forms of immunotherapy. So melanoma, excuse me, even though it's rare, only estimated to be a little over 9,000 deaths per year in the United States. Once you have metastatic disease, your chances of being alive in 5 years are only about 20%. But this medication, this interleukin-2 that turns on all the immune cells, can cure. Now we're talking patients 20, 30 years out after having this treatment without any other treatment, that their tumors never came back. So we don't use the word cure frequently, but once we're out 10, 15, 20 years, we start to use it. So that is one kind of tumor that responds to immunotherapy. Another one, for reasons that even now are still not well defined, is kidney cancer. So there are about 15,000 deaths per year in the US from kidney cancer. It has a slightly worse prognosis in that if you have metastatic disease, you have a 12% chance of being alive 5 years later. But again, interleukin-2, even at low doses, could cure patients in about 7%. And so these were the ones that we decided to investigate first. So Jim Allison was actually the Ph.D. that developed and came up with the concept of anti-CTLA-4 when he was at UC Berkeley. But he is a bench scientist, and so he did not have the resources or the partnering to try this in people. He had cured tons of mice, but he hadn't figured out yet if it would work in people. One of the things that we do well at the clinical center is translate some of the things that are risky that an academic hospital might not do. And so the company that worked with Dr. Allison to make this contacted Dr. Rosenberg in the surgery branch and said, we've got this great trial. Are you willing to partner with us to do it? And so the surgery branch actually did the phase 1 trial for ipilimumab. So ipilimumab is anti-CTLA-4. Dr. Allison, because of his work at Berkeley, joked that he wanted to call it hippilimumab, but they wouldn't let him. And the phase 1 trial got started here. So it's a monoclonal antibody. It was given at a dose of 3 milligrams per kilogram, plus a peptide that was felt to be reactive in melanoma. There were 14 patients enrolled. 2 patients had a complete and total response. All of their metastatic melanoma went away. 1 patient had a partial response. But we stopped accrual because we started seeing toxicities that we couldn't understand. So dermatitis and inflammation of the skin, colitis and inflammation of the colon, leading to severe diarrhea, sometimes perforation. Hepatitis, a near life-threatening elevation of liver function tests. And hypophysitis, which is an inflammation of the pituitary gland that affected the ability to release the necessary steroid hormones for life. So what you're seeing here is this is a lung lesion. So what we're looking at is a CAT scan. If you're not used to looking at CAT scans anytime you see them in the medical literature, what I want you to do is pretend that the person's head is behind the screen or behind the page or behind the journal article that you're reading, and their feet are pointing out at you, and they're laying on their back. So because of that, this is actually the right side of the patient. This is the left side of the patient. And we're looking at as if you were to sort of do bread loaf slices down the hole. We happen to be in the area of the lungs. I know that because I know what these shades of gray are, but I'll ask you to take my word for it. This is the heart. And just because the body is sort of a beautifully natural, symmetric thing, you can sort of see that looks like it doesn't belong there. That is, in fact, melanoma. And when you look here, it's gone. So it made a sizable tumor that's probably about 2.5 centimeters, so about 1 inch big in all dimensions, and it melted it away. But then we saw these toxicities. So this is a biopsy of skin. This is the outer edge of the skin, and these are some of sort of the follicles. What's unusual is all these little purple circular round dots. Those are lymphocytes that shouldn't normally be in the skin. So when we biopsied patients that were having this tremendous rash, what we found was that it was T cells causing the rash. Similarly, if you look here, these are the crypts of the colon. This is a biopsy of someone's colon in the midst of this colitis. All these little purple dots. And down here, this is a stain for CD3, which is a T cell marker. These aren't normally in the colon. So as this patient was having active diarrhea, bad colitis, it was T cell mediated. And again, this is a biopsy of the liver of a patient that was having bad hepatitis. All of these eventually recovered. But all these small little purple dots, again, are lymphocytes, which don't normally exist in the liver. So as drug development has always been, when you hit a toxicity, you back up. That's just the way phase 1 trials have always worked, whether it's for a blood pressure medication or if it's for an antibiotic. If you hit, and it's even named that way, dose-limiting toxicity, you back up to the dose level before and try again. So that's what we did. So we cautiously proceeded after we took, a lot of biopsies, sort of figured out what we thought was going on, knew that we understood what we were seeing, went down to a dose reduction. So instead of doing 3 milligrams per kilogram, we took it down to 1 milligram per kilogram. But what we started to find out was that patients that were responding were also the ones that were developing these autoimmune events. So it wasn't like in hypertension, when you're trying to treat high blood pressure, you can give a pill to lower the blood pressure, and the side effect is like a swelling of the throat. Those two things aren't connected in any way, shape, or form. Or you can give tamoxifen to women with breast cancer, which reduces their risk of it coming back, but the side effect is a blood clot. Those two things aren't related at all. But what we were seeing was that when we could unleash the immune system enough to destroy a tumor, it was also destroying normal tissues. And so those were linked. And so we had to make a plea to the FDA to think about this drug and this class of drug as something new and something novel. And you know how hard it is to get the government to start thinking about anything new or novel. But we convinced them to let us actually go up on the dose. But this is the data that prompted that. So we rate our adverse events on a scale from 1 to 5, 1 being mild, 5 being death. So if you can imagine maybe someone who's got a mild diarrhea that improves with just changes in their diet is a grade 2. Someone who requires oral steroids is a grade 3. Someone who requires IV steroids to the point where perhaps they need an operation to fix their colon, that's a grade 4. And someone who dies from a perforated colon, that would be a grade 5. Obviously, we didn't want any grade 5s, but when you saw patients that had a grade 3 or 4, they had a 36% chance of responding versus those that did not had a 5% chance of responding. Same with kidney cancer, 29% chance of responding, no responders if they didn't have at least a grade 3 AE. So it was with the strength of this data these data that we were able to convince the FDA that instead of going backwards, we actually needed to get smarter about how to treat the side effects, and we actually needed to go up to try to improve the response rate. So we did a formal intra-patient dose escalation, and so patients would start at 3 milligrams for their first course, go to 5 milligrams for their next course, go to 9 milligrams for their next course. And again, enterocolitis was the most common. It did happen, 18 to 28% of the time, but we could treat it with steroids. No patients died during this study. And what we found, because you might think the whole concept of steroids is to slow down an immune response, is to shut it off. Patients who get kidney transplants or liver transplants are often on a lot of steroids, and that's to shut down any sort of response. When we did this, though, it didn't stop patients who were having a response. So you had a grade 3-4 immune-related adverse event. This was a new term the FDA had to come up with. If you had a grade 3-4, you had a 28% chance of responding. Even if you only had a grade 1-2, you were still doing okay. It was if you got no immune-related event, meaning that the drug wasn't turning anything in your body on. It wasn't attacking normal tissue. It wasn't attacking cancer. But it wasn't doing anything. And so that's where we ended up in melanoma with a pretty significant p-value for response. So what we did was to try to develop algorithms for how to manage this, a very clear, stepwise manual that could be provided to providers who were going to give this drug out in the community, how long to be on steroids, when to start tapering them, what to do if steroids didn't work. Because if you could get someone, quote unquote, cured of their cancer and you could manage all this with steroids, it's a worthy trade-off. And then by looking at things years later, this trial started in 2001, and so it was updated in 2012. And what's important when you look at these trials, unfortunately in cancer, very frequently, all of them have this very steep curve at the beginning, which means a lot of people die in the beginning. But what's different about these checkpoint blockade drugs, and for the first time people were seeing in cancer, is that it levels off. There's a tail, meaning that there are, if you average it out, 20% of patients that are alive 6, 7, 8 years, unheard of for patients with metastatic melanoma. And so this went on to a bigger study on the basis of this. And that was sort of the end. Once it moved to the bigger study was when we stopped studying it here at the NCI, because our job is really early phase 1, early phase 2. Once it gets big enough for a company to think about trying to move it to market, that's really when we take a step back. So just to review again a little bit, this is the T cell. This is either the tumor or an antigen-presenting cell that's giving this little bit of antigen in its MHC. These are the co-stimulatory molecules that can be there. This T cell gets engaged. CD28 is positive, CTLA4 is negative, PD1 is negative. So these are these drugs, anti-CTLA4. It fits right in this pocket. It prevents B7 from engaging with CTLA4 and allows it to engage with the positive co-receptor. And again, anti-PD1 fits in this groove and prevents PD-L1 from latching in. These are the drugs. This is ipilimumab. It's been FDA approved. There's another one, tremelimumab, that's used in research, but has not been approved for any indication. And then nivolumab and pembrolizumab. We'll talk a little bit later. There is combination therapy with nivolumab and ipilimumab, and the only reason that combination is used is because the same drug company made both drugs. Certainly pembrolizumab and ipilimumab would work just as well as the other combination. So it left here and went into a big trial. I don't know if you can see the numbers here, but they actually treated close to 700 patients. That's volume that the clinical center would not be able to keep up with. So this was done in a phase 3 trial across the country. And randomized phase 3 means the patients don't know what arm they're going to be on. So they get either the drug ipilimumab or they get saline mixed to look like ipilimumab, so it's completely placebo controlled, and then with or without a vaccine. What they found was that you could have an objective response rate of 7%, and what they called a disease control rate, meaning that it kept your disease stable. It didn't necessarily make it go away, but it also didn't let it get bigger. A disease control rate of 22%. And on the strength of this, it was approved for metastatic melanoma in 2011. It was the first checkpoint blockade to get approved. They then updated this about 5 years later. And again, there's that tail of the curve. So even though a lot of people aren't getting a lot of great benefit from it, there's 20% of them that are likely cured of their disease. Once a line gets this flat, it generally doesn't drop too much. So 3-year overall survival, if you remember when I talked about melanoma before, at the time that this trial was being done, 5-year survival for melanoma was about 5%. So to have 20 to 26% alive at 3 years was a significant improvement. Tremelimumab, I won't belabor this, it was the other one. It just didn't do as well, and so it was not approved. But it's being used by various investigators, including, I believe, some groups here at the NCI. Nivolumab is the next big drug, and it is an anti-PD-1 drug. Their phase 1 dose escalation was done through a group at Johns Hopkins. It started off with a very low dose of 0.1 milligrams per kilogram, but then did escalate up to 10 milligrams per kilogram. These kind of plots, if you're not used to seeing them, are what we call spider plots, and they will start with, it's the change in the patient's baseline tumor. So every patient starts at 0 because there's no change from their baseline at their baseline. And then anything that goes up means they've increased. Anything that goes down means they've had a response. Anything that hits the bottom is a complete response. And so this is just one example. This is a patient with non-small cell lung cancer. And you can see, because a lot of people will say, well, maybe the disease was getting smaller anyway based on the treatment that they had before. How can you know? So this is actually a scan before the patient's treatment, and I apologize, it's a little small, but I think you have it in your handout. Again, patient's head is behind you, their feet are coming out at you. This is the right side, this is the left side. There's just this little smudge here and this little smudge here. That was before the treatment. At 2 months, it actually started to grow a little bit, but then you gave it some time, and now it's now gone again. So sometimes it takes a while for what you're seeing. The question is, at this 2-month mark, what are you seeing? Is that tumor growth, or is that T cells infiltrating and making it look fluffier and bigger? Is that an inflammatory response? Without biopsying it, to be honest, we'll never know. And for this patient, to be honest, I don't think they care, as long as their tumors are gone now. But they did look at a variety of histologies. Melanoma, 28%, non-small cell lung cancer, 18%, kidney cancer, 27%, castrate-resistant prostate cancer and colorectal cancer had no responses. That pattern has stuck with most of the other drugs that you'll see, and that these 3 tend to be responsive, and these tend not to be. What was interesting with nivolumab in comparison to ipilimumab is that there were fewer grade 3-4 toxicities, even though they had similar response rates. So perhaps it's a gentler drug because it's not engaging all these sort of self-reacting T cells that are floating around. So that was just the phase 1 trial. It required a bigger trial to get approved. And so these were patients, because ipilimumab had already been approved as a drug, patients could only go on this trial if their disease had progressed. So ipilimumab had to have failed to control their disease before they could go on to this study, and then they were randomized to either nivolumab versus chemotherapy. Now, this isn't a double-blinded trial, because when you give people choice of chemotherapy, they have to know whether the patient's getting the drug or not. So it's not the highest level of evidence, but it's as close as you could get in this era. And so 32% response rate with 4 complete responders, and in the chemotherapy arm had a 10% response rate with no complete responders. This type of plot is another way to look at clinical data, although I think it's a little bit more misleading than the spider plot. Because if you go back to spider plot and picked either the highest point or the lowest point for each patient, that's what this represents. So you don't get to see things across time. You're just seeing what the maximal or worst response is. And so the patients that did the worst, this patient went more than double. These two patients went more than double what their baseline tumor was. These patients all went, the dashed line is what's considered a partial response, which is a 30% decrease. And then these patients here had a 100% decrease. The little asterisks indicate that it's still ongoing. So these patients progressed, these patients responded. But what you can see is some of these don't have asterisks, which means even though they got down here to about 50%, at some point they started to progress. So that's why I think the waterfall plot hides a little bit and the spider plot is a little bit better, even if it's a little messier to try to see. But based on this, it was approved for refractory melanoma in December 2014. Well, they wanted to look at, well, does it have to be refractory? Maybe we can try to give it a little bit earlier. Who wants to give ipilimumab anyway? It's got this risk of giving everybody colitis. So they had to do a trial, and this was untreated metastatic disease. BRAF was another drug kind of being worked on in a parallel world, and so BRAF inhibitors had been approved. And so in order to make sure that patients were getting what was the approved therapy at the time, anyone who was not eligible for the BRAF inhibitors, which include these wild-type BRAF patients, those were the ones that were eligible for this trial. Again, this was nivolumab versus dacarbazine, which is a chemotherapy. And you can see the objective response rates there. And what I can tell you is that even though these two patients had a CR, they weren't durable. And so everything went away, but then everything came back very quickly. And so because of this, it got the additional indication of being able to be allowed for patients who had not yet been failed by ipilimumab. So you no longer had to go through ipilimumab first to get your PD-1 therapy. You could get anti-PD-1 therapy. But this was remarkable. So 5-year overall survival, 34%. Again, it's hard to sort of convey what a radical difference the world of melanoma has had in the last 10 years. Dacarbazine was approved for melanoma in the 70s. IL-2 was approved in 98, and then nothing new until 2012. And now we've got patients out 5, 6, 7 years. So it's really a phenomenal sea change. Pembrolizumab is just the other cousin made by a different drug company. And again, they started just the way nivolumab did, which is in ipilimumab refractory patients, and had great response rates and disease control rates. And again, all these references are in your handout, so if there's anything you want to look at in more detail, you can. Again, just another study of the same group. But what that led to was Pembro being approved. And then what they wanted again was to get that key first-line therapy. Can we give pembrolizumab without giving ipilimumab? And so what they tested was 2 different dosing strategies of pembrolizumab versus ipilimumab, a huge number of patients respectively. If you think about this, to do a clinical trial that captures about 10% of all the patients with the disease in the country is pretty impressive, because there's about 9,000 patients with metastatic disease in the US. And so to be able to get 834 of them onto trial, this was a pretty big accomplishment. So you can see the response rates here. No matter how frequently you gave it, either 2 weeks or 3 weeks, it's about 30%. And ipilimumab, 12%. To the credit of the drug company, they did decide, even with these results, to go with the every 3-week dosing. Some cynics would say they would have chosen the 2, but they did choose the 3-week dosing. And these are their survival curves. And again, even though the follow-up is short, so don't always get impressed by a curve like this until you look at the bottom. This is just months. This isn't years. So yes, it looks impressive, but we're going to want to see what it looks like 2 years from now. But again, they saw the same thing, which is that fewer incidences of colitis, fewer incidences of really problematic things, 10% versus 20%. So it still remains pretty good, but again, we're still only at 24 months. So we want to keep watching this data, these data as they mature. So checkpoint modulation is just another way of showing the same thing, but we've talked about all these positive and negative signals. And the two approved antibodies block here and block here. So sort of naturally what came up was, well, what happens if you block them both? And so, of course, the medical community decided to try that. And so, again, they took patients who had never been treated for their metastatic melanoma, put them in a phase 3 randomized trial. They were randomized 1:1:1. They either got nivolumab or ipilimumab or the combination of the two. One interesting thing about this is because they were trying to get a population that it might have some effect on, what they defined as PD-L1 positive was having more than 5% of the cells on just a regular pathology slide stain for PD-L1. Now, they didn't use this as an inclusion criteria, but they will break their results out by that later. And what you can see here is the percentage of patients with a response. So 44% for Nivo, 58% for the combination, and 19% for ipilimumab. And that seems pretty good, right? But so then the question is, what comes along with that? I mean, 60% response rate for metastatic cancer is pretty unbelievable. Well, this is what came along with that. The combination had a 55% chance of a grade 3/4. And so some patients choose to take this risk. Some doctors choose to take this risk. Some don't. So I would tell you that out in the community, some people just give Pembro, some people only will give the combo. And it's really just what they think that benefit ratio is. Not super important, but it didn't seem that being PD-L1 positive made much difference. If you had a high level of PD-L1, perhaps the combo makes more sense, but without it, it's not really statistically different. This is just updating. The orange is the combo. You can see the orange and the blue, not much difference, but clearly they're both different than the green, which is the ipilimumab. And this combination was approved. So why do we think melanoma works? Well, this chart lists a bunch of different tumors, and through work with the Cancer Genome Atlas, they've looked at the number of mutations present in each of these tumors. And you can see by far melanoma has the highest median number of tumors because this is a log scale. So even though it doesn't look like it's much bigger than that, it is. And so maybe it's the fact that there are so many mutations that at some point that tumor changed just enough that the body said, wait, you're not self anymore. And so it engages in non-self and builds a T-cell repertoire that can go after it. That's not been proven, but that is the current dominant hypothesis. So highly mutated tumors, we know that non-small cell lung cancer, the statistics are there, and we know there can be a correlation between smoking and mutations. Bladder cancer, just because of the nature of how it comes up, every sort of bad thing that we put in our bodies that goes through our kidneys ends up in our bladder, so it has the chance to get mutated, and it becomes highly lethal once it's metastatic. And then a different class of tumors where it doesn't actually matter what kind of tumor you have, but if it has this mismatch repair deficiency, it leads to more DNA breakage that can't be repaired, and so it leads to another, a significant number of mutations, and it's these four that people look at the most. So I'll just go through this quickly, but what you need to know is that it has been approved on the basis of sort of survival curves that look like this. Everything works at about a 20% response rate. Both squamous and non-squamous, small cell lung cancer. Again, all the details are there in your handout. I'd rather get to some of the more interesting things. Pembrolizumab, again, the same. a low volume of adverse events, but they did have some dyspnea, pneumonitis, so an inflammation of the lungs, including one that did cause a patient death. So it's something you have to be careful of. It did seem as though former or current smokers had a higher response rate, the thought being that a smoker has more mutations from the carcinogens in the smoke. So bladder cancer, anti-PD-L1s, that's the other side of the PD-1. This is called atezolizumab, and 43% response rate with very manageable AEs. And this was approved for urothelial cancer May of 2016. Pembrolizumab, similar things. Nivolumab, similar things. Although the rate of AE is a little bit higher. But 5 patients with a complete response in metastatic bladder cancer is unheard of. But this is the one that's really captured the attention of a lot of people, and that's because they were specifically looking at mismatch repair deficiencies. So colorectal cancer is often tested for this to be certain that patients don't have a familial syndrome called Lynch. And so they looked at 3 cohorts. And you would think that maybe they colored these because they went this way, but they actually colored them before they made the plot, and the colors just separated nicely. So you almost never see something like this. Anything that is red means it has its mismatch repair processes intact. It can fix DNA damage breaks. There's no problem there. It doesn't generate a lot of mutations. Anything in blue is a colorectal cancer that doesn't have that mechanism. It's been broken somehow, so it can't fix all the DNA damage, so it has a lot of mutations. And the ones in black are patients that have sort of that familial syndrome, that Lynch syndrome, that develop things like endometrial cancer, ovarian cancer, and those will have a lot of mutations. So what's really intriguing is that with a couple of exceptions, almost all of the red bars go up and almost all of the blue and black bars go down. And it didn't matter what kind of cancer it was. It could have been colorectal, it could have been endometrial. And that was really intriguing. And so they looked, and in the MMR deficient patients, you would see this interaction of PD-L1 this is tumor and this is normal, but you would see it at this interface. Lots of T cells showing up at the interface. Whereas in ones that were proficient, these brown dots are missing. So the T cells aren't rushing to the edge of the tumor in the normal tissue if there aren't mutations there to attract them. Again, this is tumor infiltrating lymphocytes. So this is at the edge. This is deeper into the tumor. And you can see there are very few lymphocytes that have been drawn to those tumors. So this approval was interesting because it's the first one the FDA has ever done where it doesn't matter what kind of tumor you have. You could have an endometrial cancer that's mismatch repair deficient and you can qualify. You can have a bile duct cancer. You can have a stomach cancer. It's the first indication that does not list the kind of cancer that you have. Most FDA approvals are approved for use in metastatic melanoma or approved for use in kidney cancer. It doesn't matter what kind of cancer, as long as you have this particular genetic test, and it's the first time the FDA had ever done that. This is just more of the same. And this is just showing that Nivo can do it too. So they're cousins. Whatever one does, the other one wants to show they can do. Sometimes one will do it first and the other will follow, and vice versa. So this is where we are. This is where it's been approved. Can we use it in these others? And the short answer is yes. I'll get to the ones that are scientifically interesting. So for Hodgkin's lymphoma, remember I said some cancers will upregulate that PD-L1 constitutively. What they found is that for some reason in Hodgkin's lymphoma, they all put PD-L1 on the surface. And so if you treat them with an anti-PD-1, they put 7 patients into complete remission for chronic Hodgkin's lymphoma. And so it got FDA approval for this indication. Again, they're cousins. Nivo did it first, Pembro did it next. Head and neck cancer, same thing, approved in August. This one is an interesting one scientifically because Merkel cell is actually created by infection of a virus. So there's polyoma virus that makes a Merkel cell carcinoma. And because it's an inflammatory response that creates the cancer, somehow blockading that response makes the cancer go away. So I don't know that they've elucidated all the reasons why, but to have 8 complete remissions in this very rare cancer that had no good treatments. Again, I keep saying things like incredible and amazing, but to be in this field in the last few years, it really has been to watch these things come out. So breaking the pathway, this is just a summary of sort of all of the things that we've looked at. And these are the ones that have been FDA approved. That I showed you. These are some that have been FDA approved that I had not yet shown you, but you can see that this chart is sort of starting to fill in. Durvalumab is being used in a lot of research currently. Avelumab was not on this original chart from Lipson, which is why it's got a little asterisk, but it's also being studied. So what about other cancers, cancers that affect more people? So this is just a sampling of the drugs that were being tested in metastatic breast cancer. Not as impressive as some of the others, but that's not stopping people from trying. Very few complete responses. Gastric cancer, again, a very small cohort. But just to show you what is being studied, so not only using these drugs in all these different cancer types, these were presented at ASCO in the last few years, it's being studied everywhere. To some good effect, to some not so good effect. So if you look at currently accruing trials in the United States as of last week, when I pulled this together, there were 585 different trials targeting either PD-1 or PD-L1, 95 targeting CTLA-4, and then 2 other checkpoints that we haven't really talked about called LAG-3 and TIM-3 because they don't have any clinical presence yet, but there are 22 and 14 trials. And the current trend, most of these, is to combine checkpoint blockade with chemotherapy. The thought that if you kill the tumor cell with some chemotherapy, it'll unleash some sort of immune reaction that checkpoint blockade can kick into. So this is checkpoint modulation. Again, anything that's red is a negative. TIM-3 was one we didn't talk about. LAG-3 was one we didn't talk about. But this is the focus of the work that will be being done in the labs in the next few years. And with that, I'll open it up to questions. So there's a lot of, the question was, is there a test to see whether patients are responding, have a higher chance of responding? And there are some tests out there for a biomarker. It has been suggested that it should be used for patients with bladder cancer. So you can test the tumor for PD-L1, and if you have a positive result, and that positive is defined based on the different antibodies, some consider that 1%, some consider that 5%, the most stringent will consider that 10%. But there's as much work as there's being done in testing the combination of chemo with these checkpoint blockade, as much work is being done in trying to develop a biomarker, because everyone recognizes that the side effects can be serious, and so they don't want to expose people to that sort of side effect if they can come up with a better way to choose their patients. Bladder cancer was the only one that PD-L1 seemed to be a more perfect, quote unquote, way to delineate between those patients, because those patients tend to be a little older, tend to be a little sicker. If you get one of those really bad ones, you're not going to come back from it. So they're trying to be a little bit more stringent in who they choose. But the search is on for the perfect biomarker.
This is a very hot area, but there's so many clinical trials. Some people say there's not enough patients. What do you think about that?
I would agree. I think that we have entered an age where I think some people, if they could, would put Pembro or Nevo in the drinking water, just like we did with fluoride. I have heard some people say that no patient with cancer should die without having gotten at least a dose of checkpoint blockade, and I think that might be a little too far to go. I think that what really needs to be discerned is the novelty. So is there a real reason why pembrolizumab in combination with cyclophosphamide should do anything, or is it just another thing to test? Some of them have very strong scientific hypotheses, and like anything in life, some are better than others. But the background of trials is always critically important to read so that you can understand why they're doing it. Because if they don't really have a great rationale, you can sort of tease that from reading the background, because it'll be like, oh, well, we think if we can kill the cell, then something magical will happen with the immune system, and then we'll ramp that up. That's not so much a scientific hypothesis as it is sort of wishful thinking. So you can sometimes tease that out, but there are a lot of trials at the moment. So it's not so much off target, it's that with CTLA-4, with ipilimumab, what you're blocking is that first initial look at an antigen. And so that could be normal thyroid or normal pituitary or normal colon. And by unblocking, this is where all the double negatives get in, where by unblocking that, you allow a cell that knows how to attack that free range to roam. With PD-1, with pembrolizumab and nivolumab, that mechanism of action is actually happening at the tumor. So it's not just at a central lymph node or at a central place. So the possibility of getting more normal tissue reactivity is higher with the ipilimumab, which is why the combination therapy had such a high incidence, because you've got not only what you're engaging at the tumor, but you add to that what you're getting up here. And in the more structured lymphoid structures, like the spleen and the lymph nodes. 56% grade 3/4 toxicity isn't anything to sneeze at, which is why some people still just use single agent, because they're scared of the double agent. And so it's the synergy of the two working together that works not only to go against the cancer, but it works to unleash more sort of normal attacking T cells out into the environs, unfortunately. All right, thank you guys.